Human cognition relies on a long-term information store that is effectively unlimited. This system allows us to retain a massive library of organized knowledge. However, there is a striking contrast between the scale of our long-term memory and the narrowness of our working memory. Working memory is our only conscious interface with the environment, yet it struggles to process more than a few nominal elements at a time. This severe limitation on how much new information we can handle at once raises a fundamental question about our cognitive architecture. Why would such a restrictive bottleneck evolve? To understand the reason for this constraint, we have to look at the parallels between human cognition and biological evolution. Both human cognition and evolution by natural selection are natural information processing systems. Because they both must solve the same problems of obtaining and storing information, they may follow a shared set of structural principles. Examining the symmetry between these two systems provides a framework for understanding how the human mind actually acquires new knowledge. When any natural system encounters novel, unorganized information, it lacks a pre-existing central executive to provide order. Without instructions, the system must default to a procedure called random generate and test. It essentially makes blind guesses about how to physically organize the incoming data, rapidly testing each configuration for its overall effectiveness. We can see the danger of this approach through simple math. Consider a system attempting to process exactly 3 novel elements. By the logic of permutations, 3 elements create 6 possible ways they can be combined. This is a manageable number. A natural processor can evaluate 6 permutations without failing. But if we increase the input to just 10 novel elements, the math changes drastically. 10 elements create over 3.6 million possible permutations. This is a combinatorial explosion. Attempting to randomly test millions of combinations would instantly overwhelm and crash any natural information processor. To prevent this, natural systems operate under the narrow limits of change principle. They must strictly limit the amount of novel information they ingest at any one time to ensure they can process it successfully. We see this principle clearly in biological evolution, where microscopic cellular structures must actively defend against constant environmental novelty. In this system, the DNA-based genome serves as the long-term store for organized biological information. Between the environment and the genome sits the epigenetic system. It acts as an intermediary, regulating the flow of information. Triggered by environmental conditions, the epigenetic system dictates exactly where and at what rate genetic mutations occur. It enforces the narrow limits of change by ensuring that most genetic information is reproduced precisely without modification. Because random mutations are rarely adaptive, they must be restricted to microscopic steps to avoid catastrophic failure of the organism. Without this epigenetic bottleneck filtering environmental novelty, the genetic code would be subject to too many random changes, and the species would fail to survive. This same architecture is mirrored in human cognition. Long-term memory is our version of the genome, a vast storehouse of highly organized information. Parallel to the epigenetic system, we have working memory. It is the gatekeeper for all sensory input. Working memory is the primary structure that processes all incoming, novel data from our environment. It is strictly constrained. When dealing with novel information, it can only process about 2 to 3 interacting elements at a time. It is also limited by time. Without active rehearsal, new information is lost from working memory in about 20 seconds. These severe constraints act as a structural defense mechanism. They prevent the brain from falling into a combinatorial explosion when faced with unfamiliar information. However, these capacity limits change dramatically when the system deals with familiar information. This is the environmental organizing and linking principle. It governs how systems function when the information is already organized. In biology, a single environmental trigger can cause the epigenetic system to activate massive, complex blocks of pre-organized DNA to determine an organism's physical traits. Cognitively, when we see a familiar cue, like an algebraic equation, working memory retrieves a massive organized schema from long-term memory. In this state, working memory has no known capacity or duration limits. The difference lies in the source. If the environment forces us to absorb novel data, we are bottlenecked. But if the environment acts as a trigger to retrieve stored knowledge, our capacity is effectively unlimited. This means we are constrained only when inputting novelty, while our ability to execute familiar, organized behaviors is unbounded. This architecture provides a structural explanation for the necessity of instructional design. We evolved to automatically acquire biologically primary knowledge, like recognizing faces or learning a first language, without conscious effort. But biologically secondary knowledge, like reading or mathematics, is novel to our evolutionary history. To learn these, we must push them through the working memory bottleneck. If instruction is poorly designed, it forces the learner to rely on random generate and test. This quickly exceeds working memory capacity and prevents learning. This brings us to the borrowing and reorganizing principle, which describes the biological basis for direct instruction. Effective instruction bypasses the need for random guessing. It takes an organized schema from the teacher's long-term memory and transmits it safely through the learner's narrow working memory to be stored. By respecting the limits of our evolutionary architecture today, we allow the learner to build the vast, organized schemas they will need to function effortlessly tomorrow.